Welcome back to the lecture series, and in the previous class we concluded with glucose biosensors, and we traveled the whole spectrum of biosensing, starting from how to use or how to process urine sample, blood sample, sweat and other body fluid to measure glucose, which are primarily invasive or minimally invasive process, because when you are drawing blood it is an invasive process, but when you are collecting urine, of course, you are dependent on the subject, yet it is a kind of a non-invasive method, but still you are completely dependent on the fluid excreted out from the body, or same is the case of the sweat. Followed by that, we moved on to non-invasive sensors. We talk about wide range of spectroscopic methods, which included Raman, NIR, talked about SPR, and we talked about photoacoustics spectroscopy, talked about thermal emission, talked about tomography, and finally, we discussed about how these data are being processed and what are the current acceptable limits of glucose sensing. So, today we will take a stock of the situation and we will again go back to the drawing board to summarize the stuff and what are the newer things what we are going to discuss about it. So, this will bring us to the world of biohybrid biosensors. So, when we talk about a typical biosensor, it has a bioreceptor that is specifically bind to the analyte. Analyte will be the analyte of your choice, it could be anything. An interface architecture where a specific biological event takes place and gives rise to a signal. And third part is the transducer element. That transducer signal, which could be anything from incoming angle of a laser beam to the current produced at an electrode, is converted to an electronic signal amplified by a detector circuit using appropriate reference and sent to for processing. So, computer software to be converted to a meaningful physical parameters describing the process being investigated. Finally, the resulting quantity has to be presented through an interface to the human operator. Biosensor can be applied to a large variety of samples, including body fluid, food samples, cell culture, and used to analyze environmental sample. So, you see that when we talk about the definition of a biosensor, these are the building blocks of a biosensor. And if you pictorially try to visualize, pictorial visualization, that is in the next slide. If you go, here is the pictorial visualization of elements of biosensing. So, these are the samples. It could be a sample from cell culture, it could be a human sample like blood, urine, saliva, it could be a food sample, it could be an environmental sample like air, water, soil, vegetation, anything and everything. And of course, while you are looking at it, what is the compound you are looking at it? That compound is your compound, say X. Now, you have to use a biological element in order to identify it. So, it could be a nucleic acid, it could be a cell, it could be an antibody, it could be an enzyme. These are broadly. So, we have talked about enzymes. If you remember, when we talk about glucose oxidase, we talked about antibodies and nanobodies. Okay. We have talked about little bit of the nucleic acid hybridization. What we haven't talked about is this part, this cell based one, and in the other aspect of it, we have talked about the whole human body. That is when we talked about all the in vivo or non-invasive sensing using different kind of spectroscopic tools. So, if you now in the light of this see these pictures, these will make more sense. Say for example, you have the minimally invasive anode implanted in your body, a cathode implanted in your body, and you are measuring glucose. Similarly, if you look at something like this, where in the earlobe you are doing the measurements. So, you have the device, you are doing not doing any kind of invasive methodology, yet you are measuring the glucose. Similarly, if you look at photoacoustic spectroscopy, where you have a laser beam and you have the ultrasound waves, and the changes out here is being monitored using a piezoelectric transducer. So, if you look at all of them in the light of this picture, it will make perfect sense that how these elements are being integrated. Now, this part 2 is the electrical interface. This electrical interface could be electrodes, nanoparticles, nanowires, FET devices, and so on and so forth. These are mostly. So, FET is the field effect transistors, devices, nanowire devices, and the signals they are picking up is signal amplifier, signal processor. So, here is the amplifier is picking up the signal, it is being processed and it goes to the display. So, this is primarily when we are talking about electrical interface. So, our journey will be now world of biohybrid biosensors with electrical interface. And in this section, we will talk mostly about whole cell biosensing. Or whole cell biosensor. That means, we will mostly talk about this group of elements, okay, which are whole cell. Now, before I moved into that topic, in order to construct a successful biosensor for non-specialist market, a number of conditions must be met. And what are those conditions? We will briefly analyze them and then we will move on to the whole cell biosensing. The biocatalyst must be highly specific for purpose of the analysis to be stable under normal storage conditions and show a long variation between assays. A low variation between the assays. Okay. So, this is the critical point. Okay. Low variability between assays. So, that means the fluctuation of result is not a healthy sign or compromises the fidelity of the biosensor. Okay. The second part, the reaction should be as independent as manageable of such physical parameter as stirring, pH and temperature. This will allow analysis of sample with minimal pretreatment. If the reaction involves cofactor or coenzyme, these should be preferably also be co-immobilized with the enzyme. So, that means the reaction should be as independent as manageable of such physical parameters as stirring, pH and temperature. That means, when you are trying to collect signal, so this interface between or this interface, there should be minimal interrogation. You should be able to pick up signal without much problem. Okay. Third point, the response should be accurate, precise, reproducible and linear over the concentration range of interest. without dilution or concentration. It should also be free from electrical or other transducer induced noises. So, when we are doing these kind of measurement, one has to understand most of these devices come with their own set of noises. So, if the noise of these devices are more, then they will interfere with the biological signals, and this part of noise cancellation or having devices with minimal noise is highly preferable for success of these kind of devices. Okay. If the biosensors is to be used for invasive monitoring in clinical situation, the probe must be tiny and biocompatible, having no toxic or antigenic effect. Furthermore, the biosensor should not be prone to inactivation or proteolysis. I will come in depth with this particular term of tiny. So, that means miniaturization is feasible. Further, it should be biocompatible. Having no toxic or antagonistic effect or antigenic effect. Furthermore, it should be not be prone to inactivation or proteolysis. That means the material should not cause any kind of inactivation or proteolysis. Okay. For rapid measurement of analytes from human samples, it is desirable that the biosensors can provide real time analysis. And finally, the complete biosensor should be cheaper or economical. It should be small, should be portable, should be capable of being used by semi-skilled operators. These are some of the basics. Now, since in this class we will be dealing with electrical interface, so and we will be mostly dealing with electrical interface of cell based or whole cell biosensor. The electrical interface giving a real time picture. So, when we are talking about whole cell biosensors, these are devices which are mostly used for research and diagnostic purposes. Okay. And they have a profound implication in drug, pathogen, toxin, terror agent detection. So, these are not the regular thing what we have talked about. We picked up a serum, we are doing a diagnostics. Do you have a higher glucose level or you are infected with some disease? Here we are trying to interface the cells in an in vitro setup. So, these are outside body situations we will be talking about, in vitro setup. We are now imagine a situation where what we are trying to do is, say for example, this is a subject and so we take out some specific cell, we integrate the cell on a electrical interface and monitor its activity when interrogated with toxin, drugs, chemicals and other agents. So, in a way, when we are pulling out the cell, this is the whole in vitro world. Now, what kind of cells are we talking about, and what are the favorite cell types which could be used for these kind of sensing? Now, if you look at human body is a electrically active system or bioelectrically active system, whichever way you want to explain human body, to realize that our body, if I have to classify the cells from a biosensor perspective, I will classify them into two groups: cells which are electrically active, cells which are electrically inactive, right. So, if I say human body, which is consist of billions of cells, and as you know, the hierarchy is cells, they form tissues, they form organs, organ form systems, and system integrate with each other to form the body as one unit. Okay. And here we have already talked about, we have measured from the whole human body so far in all our previous lecture, which is the in vivo. We have measured from blood, urine, sweat. Now, we are talking about when we are extracting out the cells from human body, and that is, we are talking about whole cell biosensors, or precisely whole cell, I will add one more word, electrochemical biosensor. Now, in order to understand the concept, you have to divide the cells of your body into two different categories. One category is electrically active category, the other is electrically inactive. So, our primary interest for these kind of understanding these kind of whole cell electrochemical biosensors is focused on electrically active cells of our body. Because electrically active cells are capable of generating a electrical impulse, and what these electrical impulse varies with the kind of stimulus which are given to them. So, say for example, a simple example, your nose. Your nose could distinguish the smell of a rose flower versus a jasmine flower. Okay. Now, when we are talking about these smells, what we are essentially talking about is there are volatile compounds which are present and these volatile compounds exhibit a different kind of biological signals in the form of electrical activity within your body. And this is exactly what we are going to exploit and utilize. And in our next class, we will talk about how these different unique features could be utilized to build wholesale biosensors, and which are one of the most potential tools in the world of basic research, diagnostics, drug discovery, toxin detection, pathogen detection, and terror agent detection. So, we will close the class here, and the next class we will start from how to distinguish the smells, signals, and what kind of signals are these, and what kind of tools we need to understand them. Thank you.